Welcome back. Today we're going to talk about gustation and olfaction, two special senses. So first, gustation or taste. Until recently, only four tastes were recognized: sweet, salty, sour, and bitter. Research at the turn of the 20th century led to recognition of the fifth taste, umami. During the mid-1980s, umami is a Japanese word that means delicious taste and is often translated to mean savory. A very recent research has suggested that there may also be a sixth taste for fats or lipids. Gustation is the special sense associated with the tongue. The surface of the tongue, along with the rest of the oral cavity, is lined by a stratified squamous epithelium. Raised bumps called papillae, singular equals papilla, contain the structures for gustatory transduction. There are four types of papillae based on their appearance: circumvallate, foliate, filiform, and fungiform. Within the structure of the papillae are taste buds that contain specialized gustatory receptor cells for the transduction of taste stimuli. These receptor cells are sensitive to the chemicals contained within foods that are ingested, and they release neurotransmitters based on the amount of the chemicals in the food. Neurotransmitters from the gustatory cells can activate sensory neurons in the facial, glossopharyngeal, and vagus cranial nerves. The tongue. The tongue is covered with small bumps called papillae, which contain taste buds that are sensitive to chemicals in ingested food or drink, as we said. Different types of papillae are found in different regions of the tongue. The taste buds contain specialized gustatory receptor cells that respond to chemical stimuli dissolved in the saliva. These receptor cells activate sensory neurons that are part of the facial and glossopharyngeal nerves. Salty taste is simply the perception of sodium ions in the saliva. When you eat something salty, the salt crystals dissociate into the component ions sodium and chlorine, which dissolve into the saliva in your mouth. The sodium concentration becomes high outside the gustatory cells, creating a strong concentration gradient that drives the diffusion of the ion into the cells. The entry of sodium into these cells results in the depolarization of cell membrane and the generation of a receptor potential. A sour taste is the perception of hydrogen concentration. Just as with sodium ions in salty flavors, these hydrogen ions enter the cell and trigger depolarization. Sour flavors are essentially the perception of acids in our food. Increasing hydrogen ion concentrations in the saliva, lowering saliva pH, triggers progressively stronger graded potentials in the gustatory cells. For example, orange juice, which contains citric acid, will taste sour because it has a pH value of approximately 3. Of course, it's often sweetened so that the sour taste is made. The first two tastes, salty and the sour, are triggered by the cations sodium and hydrogen. The other tastes result from food molecules binding to a G protein coupled receptor. A G protein signal transduction system ultimately leads to depolarization of the gustatory cell. The sweet taste is the sensitivity of gustatory cells to the presence of glucose dissolved in the saliva. Other monosaccharides, such as fructose or artificial sweeteners, such as aspartame, saccharin or sucralose, also activate the sweet receptors. The affinity for each of these molecules varies, and some will taste sweeter than glucose because they bind to the G protein coupled receptor differently. A bitter taste is similar to sweet in that food molecules bind to G protein coupled receptors. However, there are a number of different ways in which this can happen because there are a large diversity of bitter tasting molecules. Some bitter molecules depolarize gustatory cells, whereas others hyperpolarize gustatory cells. Likewise, some bitter molecules increase G protein activation within the gustatory cells, whereas other bitter molecules decrease G protein activation. The specific response depends on which molecule is binding to the receptor. One major group of bitter-tasting molecules are alkaloids. Alkaloids are nitrogen-containing molecules that are commonly found in bitter-tasting plant products, such as coffee, hops in beer, tannins in wine, tea, and aspirin. By containing toxic alkaloids, the plant is less susceptible to microbe infection and less attractive to herbivores. Therefore, the function of bitter taste may primarily be related to stimulating the gag reflex to avoid ingesting poisons. Because of this, many bitter foods that are normally ingested are often combined with a sweet component to make them more palatable, for example, cream and sugar in coffee. The highest concentration of bitter receptors appear to be in the posterior tongue, where a gag reflex can still spit out poisonous food. The taste, known as umami, is often referred to as the savory taste. Like sweet and bitter, it's based on the activation of G protein coupled receptors by a specific molecule. The molecule that activates this receptor is the amino acid L-glutamate. Therefore, the umami flavor is often perceived while eating protein-rich foods. Not surprisingly, dishes that contain meat are often described as savory. Once the gustatory cells are activated by the taste molecules, they release neurotransmitters onto the dendrites of sensory neurons. These neurons are part of the facial and glossopharyngeal cranial nerves, as well as a component within the vagus nerve dedicated to the gag reflex. The facial nerve connects to taste buds in the anterior third of the tongue. The glossopharyngeal nerve connects to taste buds in the posterior 2/3 of the tongue. The vagus nerve connects to taste buds in the extreme posterior of the tongue, verging on the pharynx, which are more sensitive to noxious stimuli such as bitterness. Next sense is olfaction. Olfaction or smell. Like taste, the sense of smell or olfaction is also responsive to chemical stimuli. The olfactory receptor neurons are located in a small region within the superior nasal cavity. This region is referred to as the olfactory epithelium and contains bipolar sensory neurons. Each olfactory sensory neuron has dendrites that extend from the apical surface of the epithelium into the mucus lining the cavity. As airborne molecules are inhaled through the nose, they pass over the olfactory epithelial region and dissolve into the mucus. These odorant molecules bind to proteins that keep them dissolved in the mucus and help transport them to the olfactory dendrites. The odorant proteins complex binds to a receptor protein within the cell membrane of an olfactory dendrite. These receptors are G protein coupled and will produce a graded membrane potential in the olfactory neurons. The axon of an olfactory neuron extends from the basal surface of the epithelium through an olfactory foramen in the cribriform plate of the ethmoid bone and into the brain. The group of axons called the olfactory tract connect to the olfactory bulb on the ventral surface of the frontal lobe. From there, the axons split to travel to several brain regions. Some travel to the cerebrum, specifically to the primary olfactory cortex that's located in the inferior and medial areas of the temporal lobe. Others project to structures within the limbic system and hypothalamus, where smells become associated with long-term memory and emotional responses. This is how certain smells triggers emotional memories, such as the smell of food associated with one's birthplace. Smell is the one sensory modality that doesn't synapse in the thalamus before connecting to the cerebral cortex. This intimate connection between the olfactory system and the cerebral cortex is one reason why smell can be a potent trigger of memories and emotion. The nasal epithelium, including the olfactory cells, can be harmed by airborne toxic chemicals. Therefore, the olfactory neurons are regularly replaced within the nasal epithelium, after which the axons of the new neuron must find their appropriate connection in the olfactory bulb. These new axons grow along the axons that are already in place in the cranial nerve. Disorders of the olfactory system, anosmia blood. Forced trauma to the face, such as that's common in many car accidents, can lead to the loss of the olfactory nerve and subsequently loss of the sense of smell. This condition is known as anosmia. Currently, you know that anosmia is the symptom of COVID-19. When the frontal lobe of the brain moves relative to the ethmoid bone, the olfactory tract axons may be sheared apart. Professional fighters often experience anosmia because of repeated trauma to face and head. Anosmia may also be related to some presentations of mild depression because the loss of enjoyment of food may lead to a general sense of despair. The ability of olfactory neurons to replace themselves decreases with age, leading to age-related anosmia. This explains why some elderly people salt their food more than younger people do. However, this increased sodium intake can increase blood volume and blood pressure, increasing the risk of cardiovascular diseases in the elderly. Alright, dear students, that's it for today. Subscribe please to my channel. Put comments on the comment section. Good luck and see you.